hand and interrupt with questions. I will hopefully see the hand pop up. I'm going to put the link to the slides in the chat because within the slides there's a couple of other links, and if you are interested in following up on references, then you have everything available. And I'm going to talk about how we can model uncertainties in a paleoecological context. That's just with a link to the slides. And I put a link to my website, but not for the shameless self-plug, but because there are some more resources in there. If anyone is interested, there's some new resources on how to model paleoecological data using state-space modeling, for example. So anything you're interested in following up on. So the problem we have in, well, all data, but paleoecology in particular, is that the data are the product of multiple sources of uncertainty that layer up along the way. So we have environmental processes like bioturbation or taphonomy, variable sedimentation rates in our core that like to mess up our data. We have the field and the lab methods we apply, so the core collection itself and how we subsample the core, how we quantify our proxy, whether that's pollen or diatoms or isotopes. And then we have what we do to the data themselves. Once we've got the numbers after these layers of uncertainty, then we're doing things like age depth modeling. Maybe we're smoothing our data, interpolating it, applying some sort of statistical method. And so the question that we got to keep our eye on is the past actually recoverable from the data? Because that's our objective, right? We want to reconstruct the past from our data. And paleoecology is moving toward a more quantitative discipline as well. Originally, it was a massive advancement just to be able to recreate the past, just to describe it. But now we want to quantify that change with respect to something like changing environmental patterns. There's a lot of interest in using paleoecological information to inform the future as well. What management recommendations or predictions, I use that word cautiously, we may want to make from paleoecological data. And we're also constantly advancing laboratory and statistical methods. Statistical methods, there's so many more available now. There are many proxies available now that weren't formerly available. So we've got to be able to understand how our data can actually reconstruct the past, given the uncertainties that they face. One way we can quantify uncertainties is in simulation. And the approach I'm going to describe is this pseudo-proxy modeling, or virtual ecology, we're going to call it. And the idea is that you simulate your data. We're going to simulate core samples. We're going to simulate process and observer error that affect those samples. So processes such as environmental processes, the taphonomy, bioturbation. We're then going to simulate the observer, and we're going to assess how statistical inferences are affected by those processes, process and observer error. And I'm going to go through this step by step, but the main take-home message is that proxy uncertainty, so the proxy themselves carry uncertainties, which are kind of like the counterpart to chronological uncertainty. And proxy and chronological uncertainty are not actually separate, and they can have combined effects. So I'm going to go through these key concepts one by one. We have virtual ecology, we have pseudo proxy experiments, and we have pseudo proxy system modeling. Going to get them confused myself. And then I put in some key references here as well. The first paper by Blau talks about how we can recreate statistical patterns from random walk processes, statistical patterns that are very similar to those we see in empirical paleoecological data. Paper by Williams that talks about intrinsic and extrinsic dynamics and how those can drive our proxy data. And the work that's actually stemmed off this presentation that I am giving you, which is from my PhD in New Zealand. So going through these concepts one by one, virtual ecology is a framework for assessing a sampling or analytical method in simulation. So it consists of generating a model or developing a model that generates data, I should say. That model can be as complicated as you like. It can be a process-based model that simulates every individual on a landscape, or it can be generating random patterns that you're going to sample from. So that's the first step. The second step is a simulated observational process. So now we're simulating the observer. So we have our simulated landscape. It could be, say, like the spatial heterogeneity of chemicals across a catchment, and we're going to observe from that. So we're going to say, okay, how many sensors do I need across my landscape to accurately represent the heterogeneity across it? And then we have an analytical process applied to the data. So now we have two sets of data. We have our generated synthetic data, our landscape, our proxies, whatever our study system is. So that's our like perfect representation of the landscape. Perfect in quotes because of course it is simulated. And then we have our observed data from that. And this is a simulated observational process. So now we have two data sets. We have one that is a representation of the other. And we can statistically analyze both. And the difference between the two is how much our observational process or how well it recreates the original data. And that's the fourth part, actually. That's the assessment part. I blended 3 and 4 there. And we're going to do the same thing for paleoecology. So that's the first concept. I like to think of it like this. In your simulated data, you have the perfect knowledge of an imperfect world, right? You know how you came to that world. We have known drivers, we have known responses, known environmental and observational processes, and this is like our perfect benchmark. It's our control data, and we can replicate that as much as we like, as long as we have computational power, and we're able to systematically introduce sources of uncertainty. So we can change our sampling regime, for example, and we can do this all day long. So we have that advantage of replication. But it's not perfect because we simulated everything. But then in empirical ecology, we have the perfect world, right? But we have imperfect knowledge of it. No matter what we do, we're always observing that world. We don't really, even our best data, we don't have a perfect control. Of course, the advantage of it is real. It is reality. So we're kind of playing this game between simulation and reality. The second concept of pseudo-proxy experiments, I'm borrowing the term from climatology. And pseudo-proxies are basically modified or simulated data. So very much like step 1 in virtual ecology. The core concept is that they need to mimic the statistical properties of empirical data. And it's actually very similar to virtual ecology, but I'm borrowing the term pseudo-proxies to refer to the simulated proxies, and it comes from this body of work embedded in climatology, which very well describes pseudo-proxy experiments. So I like to use both. And finally, we have this concept of proxy system modeling. And proxy system modeling is the framework that describes how environmental change is recorded as an observable signal in our proxy data. So we have some sort of environmental change, such as climatic variability. We have something that responds to that change, like a physical, biological, chemical component of the system that is going to respond to that change. Maybe it is corals or trees responding in the density of tree rings, or responding in their abundances to a change in temperature as it becomes more or less favorable. And that response is then recorded in some sort of archive or a medium that records that response. So in our case, that is like a sediment core that is recording the response in changes of abundances in, say, pollen data or diatoms. That archive could be the trees themselves, the woody matter that records the response of the sensor in the tree ring data. Could be corals as well in coral rings. And then we have some sort of model that draws observations from that archive. So this is, I think, a very important concept because we really need to be very well aware of how well coupled our observations are to the original signal. And along this process, we might be muddying that signal a little bit. So maybe the environment is really closely coupled to the sensor. It responds very rapidly to environmental change, but it might degrade in the archive. And then by the time we've extracted observations, we're no longer that well coupled between observation and what we're trying to recreate. So with those concepts in mind, I'm going to describe how we built a model to assess uncertainties. And what we set out to do is we wanted to represent multiple drivers in our environment. So we were interested in how interacting drivers might change our pseudo proxy or our simulated data. We wanted to include some underlying ecological dynamics, such as growth rates and a niche for the environmental change. We wanted to generate a multi-species record, so we're not just interested in a single time series, we're interested in multi-species analyses. We wanted to recreate something of the core formation processes, such as the accumulation rates and the time span of the data. And then we're recreating the observational process. So you can see here how we're fitting this in, or how this fits into the virtual ecology framework, as well as the proxy system modeling framework, because we have those drivers, we have the sensor, we have the and we have the observation. So I'm going to start up with what the model looks like. And this is a demonstration of the model itself. I don't know if you can see my cursor on the screen or not. Can you see my cursor on the screen? We can.
Yeah.
All right. So on the left-hand side, we have the core itself. So this is the representation of the core. It has some depth and it has some accumulation rate in this case. So it's about, and it has a time span. So it's about 2 meters thick, well, a little bit over 2,000 centimeters. That is covering approximately 5,000 years or 5,000 model time steps, and has a variable accumulation rate. So its accumulation rate over time does change, and this will affect the sampling when we do our simulated observational process. When we take a sediment layer of, say, 1 centimeter thick slice of sediment and quantify the proxies from that, it's going to have some sort of time averaging based on the accumulation rate. Then we have how our environment is changing over time. In this example, I've used a fairly simple driving environment of just a monotonically changing primary driver. And what this is going to do is it's going to ensure some sort of species turnover over the model simulation time. And then we have some random noise that is occurring during the simulation. So that's our driving environment. And then we have our sensor. Our sensor in this case is how each species is going to respond to each driving variable. So we have 2 curves here, and that's a niche for each driving variable for each species. So for example, it's going to have, each species will have a tolerance per driver, and that is going to determine the species growth rate. So if both environmental drivers fall on the optimum of the niche for a species, it's going to have its maximum growth rate, and it's going to be happy, it's going to reach its carrying capacity. But if either driver falls outside of the tolerance of that species, then the growth rate will become negative, and the species will slowly die out and only become reintroduced if conditions become favorable again. So here we have 6 example species. These are just randomly selected species that had some abundance in the simulation. So for example, species 1, it apparently is very generous over here. It seems to be surviving for almost the entire simulation until right at the end, when both drivers seem to go outside of its tolerance and it starts to die out. The next species along only actually favors those final environmental changes and comes into existence. that species is clearly very sensitive to the random noise variable because it very closely replicates this little dip in that random walk. So this is how we're beginning to build up species. So each species has a niche for each environmental driver, and that determines its growth rate. If the conditions are favorable, it grows positively to its carrying capacity. If the conditions are not favorable, it will die out. So now we have our simulated data, we've got to start to sample from it. But I've included an extra step here because we're doing this in a paleoecological context. Before we observe our data, we have processes that affect them before we even get there. And the one I've used is simulated mixing. So it's, for example, bioturbation. So we have our sort of error-free data, this simulated pseudo-proxy of exact abundances. So these are like, you can consider these as the exact number of pollen grains for that species in that core. And then we're going to muddy that signal. It's going to get mixed up over time. So we're going to apply like a centrally weighted moving average. So we're going to essentially smooth the data. I forgot to mention that these little red bars are random generated disturbances that will off-take a certain proportion from each species. So here we lost a little bit of that species and it's going to recover over time because conditions are still favorable. So now we've mixed up our data a little bit. The next step is that observational process. So now we're going to subsample the data. So this is, if you can imagine, we're taking 1 centimeter thick sediment core slices every 10 centimeters, for example. So now we've subsampled from our data, and then we're going to take those subsamples and we're going to essentially recreate the process of quantifying or counting individual pollen grains from those subsamples. So now we have all those 3 layers of uncertainty combined, and this is our observed data on the right here. So we've gone from this perfect core of absolute abundances of all the species in that core. We know how they got there because we simulated our driving environment. We know what their tolerances were to that driving environment. And then we've slowly ruined it and we've recreated the observational process. And then we start to get something that looks a lot more like what we see in actual paleoecological data. So like I mentioned, I extended this proxy system model framework to include this degradation process of processes that occur between the formation of the archive and when you take those observations of that archive. So I'm going to show a couple of images that just extend what we've looked at, the archive we've looked at, because we only saw, 6 species to begin with. We actually simulated 200 species. So we simulate a 200 potential species, and probably only roughly 30 will exist at any one given point in time. But as those driver conditions change over time, we have this large pool of species that can enter, and probably will, as others die out. So here's an example where we have our drivers on the left, our error-free data, and then what that looks like once it's been degraded, subsampled, and counted. This is 20 of those species instead of 10. So you can see that we've now built up a record that looks a lot more like what we actually see in paleoecology. This is a little bit compressed because, of course, we got 5,000 simulation time steps. So this plot should be a lot taller, which would make the features a little bit more less visually rapid. So now we have these 2 sets of data. We have our error-free benchmark, we have our observed data. We're going to analyze both sets of data and see how might our statistical inferences change once we've layered up all those uncertainties along the way. Something I failed to mention is we have the core formation, so we have a known age of those 5,000 time steps. I've also applied an age depth model to that. It doesn't come into play in what I'm going to talk about today, but we do have estimated ages that are essentially sampled radiocarbon dates throughout the core at different resolutions. I'm going to talk about analyzing just the proxy data for now, though, or the pseudo-proxy data, I should say. So we've generated the data. We're going to apply two different analyses. I'm mostly going to follow one, but I'm going to show you two just to begin with, but it gets convoluted if I try and describe too much. So this is an analysis. On the left, we have Fisher information. On the right, we have principal curves. And this is an analysis of what we just saw. It's an analysis of this and this. So on the left-hand side, Fisher information is essentially a dimension reduction method. So it's a multivariate analysis. It's quite simple. It's like a moving window analysis that for a while people thought was a great representation of stability in a system. I'm less convinced now that I've used it, that it is that particularly appropriate to paleo specifically. I think the theory is great, but as you can see, it's giving me very noisy signals, but it is recreating overall the trajectory of the system quite well. So imagine it is just a dimension reduction method of a multivariate system. So we're collapsing those 200 species into one analyzed data or one analyzed series. And on the left here is the analysis of the error-free data. On the right here of A is the analysis of the degraded data, and we're going to see how they change. For the most part, they're actually quite similar for the first few thousand years, but then things do actually begin to change quite a lot. For example, this period here that I've highlighted is about 700 years. This is where I kind of want the plots to be a lot taller. Because if we see a 700-year period of quite a sharp decline of Fisher information, that we're making inferences about the stability of the system from. If you see a 700-year decline of Fisher information, you're like, wow, this system is losing stability fast. And over 700 years is a decent length of period over which stability is changing. Whereas on the right, that period is actually increasing. So we can be changing our inferences quite a lot. If we had taken only the top 1,000 years of this core, or 1,500 years of this core, and we saw this massive decline, we would be a little bit concerned over what's happening to that system. Principal curves, you can consider them as like a nonlinear principal component analysis. So we're fitting a plane through a cloud of data, basically. And as we can see, the principal curves are actually representing that just monotonically changing linear driver over time pretty nicely. This is a good representation of how the species in the system are turning over time. But in our degraded data, we're seeing a much more like S-shaped curve. So if we degrade our data, we might actually be interpreting this visually as two different periods of species composition. So we can see that the degrading our data, well, of course, it's going to have some effect, but we want to know how much effect. So we're going to just recap that briefly, because there's quite a lot of information there. We've gone through the whole model building process. We've gone through analyzing the two data sets out the other end. So what we have is we have environmental driver patterns over time, and in the pseudo-proxy system framework, that's our environment model. We have the species that respond to those drivers, or our sensors. We have a representation of the core itself, which is done with accumulation rates, time span, and depth. We have some sort of degradation model, which in this case is core mixing. We can have many more submodels there. For example, taphonomy. We have our simulated sampling and counting process, so taking individual sediment, 1 centimeter thick sediment slices, and then quantifying the proxies from those slices. And then we have an analysis of those results, which is the assessing in the virtual ecology framework, it's assessing how those uncertainties affect the data or affect our statistical inferences from the data. And what I've shown you is one scenario. We simulated actually multiple different driving environments. I've just shown you the one with that sort of gradually changing environmental driver. I think it's quite a nice scenario because we know that by the end of the simulation time, we should have undergone a full species turnover or near complete species turnover. That was only one replicate that we looked at, and it's just a randomly pulled replicate. We simulated 30, probably more replicates actually, to account for the stochasticity in the model and the variation across replicates. And what we looked at was one level of uncertainty. We had our error-free benchmark, and then we had the one that had been mixed, subsampled, and counted. So we had our observed data set. But what we did in reality is we simulated every step along the way. We simulated 10 different levels of mixing. We simulated 10 different sampling intervals. We simulated 10 different counting resolutions, so 200 grains, 300 grains, 400 grains. And then we did all the combinations of those three. So you actually end up with a gradient of uncertainty, which is 1,210 different data sets when you account for all the possible combinations. So what we've looked at so far is a nice tangible visualization of the data, the analysis of the data, but one replicate doesn't tell us what we actually want to know. So I put in a slide here for questions. Because we covered a lot of ground there, if anyone has any questions, now would be a good time to ask about what we've just gone over. And I'm going to have a sip of coffee. All right, no questions. That's either really good or really bad. So now we've got to extend this to actually quantifying how those uncertainties affect our data across all those simulated replicates and data sets. So across the replicates, for each of the replicates, we have these 1,210 data sets, which is our gradient of uncertainty from our error-free reference benchmark to the one that's been heavily mixed, has the largest sampling interval, but say 10 centimeter sampling interval between sediment slices, and a really low proxy count resolution of like 100 pollen grains per subsample. That'll be our worst case scenario. And how we do this is we have to extract features from the data. I'm actually not going to delve into this too much because it's a bit convoluted to explain. So we're going to go with a miracle occurs, and then we're going to see some results. But I'll give you a brief overview, and I do have some extra slides at the end. If anyone has questions about the exact methods, I can go a little bit deeper if we have time. But basically, in essence, what we need to do is we need to calculate some sort of difference between our error or distance from our error-free benchmark and each one of those 1,210 data sets all the way to an end. So what we expect to see is that distance increase. So the more uncertainty we apply to our data, the further away we get from our error-free benchmark. So very briefly, this is how I tend to visualize that. So you can consider it like a three-dimensional array, where each dimension of that array is a replicate. So we have sort of replicates going 1, 2, 3 here, all the way up to 30 plus replicates, replicate simulations using the same driving environment. Each column is a layer of uncertainty. So our first column is our error-free benchmark, and then we have our one that's been mixed at essentially weighted moving average of 1 centimeter, and then all the way up to 10 centimeters. So each column, you can extend that to 1,210 columns, is a layer of uncertainty. And each row is the result of the analyses. So you can imagine that each time step here is a metric from the Fisher information. So insert in that column this image, pretty much. That's how I think of it. But again, we're not going to delve into this particularly deeply because it gets complicated fast. The idea is now from each of those columns, we need to extract some sort of distance metric to make them comparable. And to do this, we use feature analyses or time series, which is a dimension reduction method that reduces that Fisher information time series or the PRC series into a set of metrics that describe that series. And from that set of metrics, we can calculate the Euclidean distance across all the levels of uncertainty. For now, that's all I'm going to say about that. What we're actually interested in is what that means and how that affects our data or our statistical Fisher information in this case after the analysis has been done. So what we're looking at here is for each uncertainty individually, what is its effect on our Fisher information? And this is kind of what we would expect to see. The more we ruin our data, the more distant it gets from our error-free benchmark. So here we have the Euclidean distance on the y-axis. Each dot is a level of uncertainty. Each facet is that uncertainty. So for example, here we have mixing. We have the bioturbation. We have it at 2 centimeters, 3, 4, 5, 6, and then finally we have mixing over 10 centimeters in our core. The error-free benchmark would just be 0 down here somewhere. So that's not plotted. It's always going to be 0. So we can see that as we mix up our data, which is kind of like the same thing as smoothing it in this case, we're getting further and further away from our error-free benchmark. No surprise, but reassuring that the method is working. Same thing for proxy counting. When we start to quantify start to count and identify individuals from our subsamples, we see this gradual increase, although not as much as you might expect, as we count from our data. So you have that initial sort of large increase in distance, which is what you would expect. Just applying the method itself is observational error. We can cope with that. But interestingly enough, when you apply that uncertainty individually, there's not a huge difference between whether you're counting 400 individuals or 600 individuals. So how much that distance changes per uncertainty is far more interesting than that it is actually changing, because we knew that was going to be the case, right? That's we already know that you ruin your data, it gets worse. And then we have our subsampling interval. So going from a contiguous 1 centimeter resolution subsampling interval regime to gaps of 10 centimeters between samples. And this is having probably clearly the strongest effect on the data. But these are each being applied individually. So we can see the relative difference, that's useful, and we can see the relative increase within the plots. But what we are potentially more interested in is how are these uncertainties going to interact when they're applied in combination. So this is nice and simple. One by one, this is the advantage of the virtual ecological approach, is we can systematically introduce uncertainty. We can say, what happens when I mix my data? What happens if I don't mix my data, but I subsample it? So that is a useful systematic introduction. But then we can apply these in combination. So these complicated plots are what happens if we apply mixing and subsampling together, mixing and proxy counting, or proxy counting and subsampling. So it's all the possible combinations of uncertainty. And what we're most interested in here is actually the diagonal axis. So we know that the first column over here of mixing is more or less going to be the same as that. And this last row here of subsampling is more or less going to be the same as that. So what happens when we apply the two together? Are we seeing more increase in distance from our error-free core? So is there an interaction effect between the uncertainties? And in this case, I would say not too much. It's actually fairly marginal. Why that is because the uncertainty from subsampling alone is basically overpowering that little bit of smoothing we're getting from that mixing. So yeah, there's probably some small interaction effect if I really say, but subsampling is the dominant process. I'm not really seeing anything when we apply mixing and counting together. Again, I think counting that proxy count resolution is the dominant process, and just applying that process has that massive distance increase, whereas applying mixing, we start at one, we're only mixing a little bit at a time. So we clearly have a dominant process there. But when we get to applying subsampling and the proxy count resolution, clearly up on this top right corner here is the largest increase in distance from our benchmark. So here we are seeing an interaction effect. And so this is where things get more interesting. And we can do this, and I have done this for 3 dimensions. So we do have 3-dimensional plots, but I have not included that because it's honestly a bit of an eyesore to try and take that in. And we can already deduce from this that when you layer on that 3rd dimension of uncertainty, it's going to become more distant from the error-free benchmark. That's not rocket science. We know that's going to happen. But from just the 2 dimensions of uncertainty, we can see that mixing is having less effect. And our dominant interaction effect is from the interaction of subsampling and proxy counting. And in a sense, this is kind of reassuring because when we're doing empirical ecology, paleoecology, these are the 2 aspects which we have some control over. We can, in the lab, we can increase our subsampling strategy, increase our subsampling resolution and increase our proxy counting resolution. Whereas the mixing is something that happens in the environment. We have no control over that at all. Again, I put in a little break for questions if anyone has anything so far, because again, we're covering a lot of ground at a time here. Also give me a second to restock on caffeine. So how does this actually help us? So the main takeaways is it helps us understand the relative effects of these uncertainties. So like I said, it's not rocket science that if you ruin your data, your analyses get worse, but what source of uncertainty is actually having the most effect on our data, or more importantly, our statistical inferences from those data? We do very much like to eyeball patterns in paleoecology. And so our statistical inferences robust of 10 centimeter sampling intervals and a 300 count resolution of our pollen proxy or our diatoms. It allows us to consider how we might miss it for a given signal, a false positive. We can take advantage of replication. Replication is very rarely done in paleoecology. I can think of one example where more than one core was taken from a lake. So we have very poor understanding. We rarely replicate across space, and we rarely replicate within a core, right? It's not common that we do repeat counting of a sediment, 1 centimeter thick sediment slice, and try and quantify our observational uncertainty, say recount that 3 or 5 times and see what our margin of error is in the count itself. So we don't tend to know much about spatial uncertainty if we take multiple cores, and we don't tend to know much about within core count uncertainty in an empirical sense. And of course, that's because it takes a huge amount of time and effort to develop these records. This might change. I've been to a few talks on artificial intelligence methods for image recognition of pollen grains, which seem to be progressing pretty well. So perhaps we're going to be able to develop pollen or diatom, well, maybe not diatom, pollen records really well and rapidly. And it also helps us to understand the effect of proxy uncertainty on the chronological placement of events. So this is, I think, also really important and underestimated. So something I haven't shown here because it would have been a bit of an overload, but one of the results is that we came up with is that our uncertainties in the proxy themselves, not related to the building of the chronology, but the uncertainties in the proxy themselves can actually alter the placement of a chronological event. So say you have a big change in species turnover, and this is a scenario we simulated. We simulated just a big step change in our driving environment, and so we have this sort of stark shift in species turnover, and identifying the timing of that stark change in species composition could actually be misplaced as you introduce uncertainty. So we're actually changing the chronological timing or the placement of those events in time with proxy uncertainty as well as chronological uncertainty in the age depth modeling process. There was between that and the speed, the response rate of the system. So if you had a system full of generalist species, then the species would actually quite slowly go in and out of existence with respect to that stark step change in the environment. So it wasn't very strongly coupled to our environmental sensor. Remember what, environmental driver. Remember what I talked about at the beginning of how well coupled is each step of that proxy system model? Actually, I'm going to bring that image back up. So in our very slow response rate environment, or very slow response rate sensors, they weren't very strongly coupled to the environment. And then we have our degradation of the archive, and then we have our observational process, which, if in the worst case scenario, is not very strongly coupled with our sensor or our archive, to be exact. So we can quite rapidly see the issue here with these layers of uncertainty, but also how our environment and our sensors and our observations and our degradation are decoupling our ability to recreate the signal of interest. And how do we apply this to empirical situations? Because, of course, we've just talked about simulation world here, and it can be quite hard to couple simulation with perfect So this is where we're playing that game in virtual ecology between our perfect world with our perfect empirical world with imperfect observations and our imperfect simulated world with perfect knowledge. But what we can do is, before we even conduct a study in the field, we can consider what question we're actually asking. What is our signal of interest? Are we trying to pick up something like an abrupt shift that we hypothesize has happened in the environment? And if that's what we're interested in, what source of uncertainty is going to affect that the most? And in this case, for example, in that, it would be subsampling resolution, because that can alter our chronological timing of the event, along with radiocarbon dating resolution. We found that principal curves actually represented long-term system trajectories pretty well. So maybe we don't need to focus really heavily on our proxy count resolution or our subsampling resolution. And we do want to create, say, replicate our cores in space. because then we can recreate that long-term system trajectory. We can have some confidence that the principal curve analysis is going to do that fairly well. And we have three replicate cores to do that across, even if they're sampled at, say, 10 centimeter intervals at a 400 count resolution. Actually, are we getting that long-term system trajectory, and our trade-off being a reduction in subsampling resolution, but an increase in spatial resolution, of course. I kind of covered points 1 and 2 there. Basically, we're trying to increase the likelihood of detecting a hypothesized signal in the data. And we can also accompany empirical studies with a simulation counterpart. So we can have our empirical data, and we can also simulate what we think is happening as a topic in itself. The simulation I've shown you has been relatively simple. We're recreating patterns that mimic the statistical properties of the data. What we haven't done is recreated mechanistic processes that lead to those patterns. And then we can assess how robust our inferences might be given the data and given the uncertainties. And these are things that are really, well, commonly talked about, but rarely done in paleoecology. And this demonstration is just one method you can use to begin to quantify uncertainties in simulation. So there is of course, there's always an outlook. There's assessing error rates, and this is something we did do actually. So this isn't really an outlook. So we, across the replicate simulations, we did an analysis, which was how many simulations across the replicates would correctly identify a change point in the data. And then you do that across the levels of uncertainty, and of course that the proportion of simulations in which a change point is correctly identified reduces as you increase the uncertainty. And that's also how we came to the result of the identification of that change point changing in time a little bit as you introduce uncertainties. But the really cool extension, which I haven't done, which I think would be very interesting, is to combine that chronological and proxy uncertainty. So if you recall, I mentioned that this model, we do actually have a age depth model accompanying it. So from our core representation, we took radiocarbon samples or simulated radiocarbon samples at different intervals. So we can recreate our age depth model. So we have chronological uncertainty, and we can combine that with proxy uncertainty. And then, of course, we can always extend the underlying dynamics. So this model has it is completely reversible. There might be a time lag, but if you reverse the environmental conditions, eventually the same species will come back into existence. It might just have some time lag in that, which is not the same thing as true hysteresis, where for different driving environments, we may have two completely separate species compositions that could potentially exist given the same conditions. So we can always extend the underlying dynamics to test different things. Maybe we want to test bistability, then we need to change our underlying ecological equations. The final thing to remember is that, of course, all models are wrong, but some are useful. So hopefully this model is useful, but it is absolutely wrong. In the sense that we have, for example, a relatively simplistic representation of core mixing. We have a relatively simplistic representation of species niches. So we know that the model is imperfect, but it, of course, generates us perfect knowledge. So any final questions? That brings me to an end. I may have gone through that a little bit. Oh no, I'm pretty okay for time. Actually, we have 5 minutes for questions. I was trying to hit the sweet spot in the caffeine. That means I don't speak too fast, but I still get through all the information. So, of course, acknowledgements. My supervisors, PhD supervisors in New Zealand, George Perry and Janet Wilmshurst, who fantastic George Perry is an amazing simulation ecologist, and Janet is an amazing empirical ecologist. So we really did manage to combine a lot of expertise in this. And then Jack Williams and Tony Ives, honorable mentions from my postdoc over here at UW-Madison, because again, just incredible wealth of knowledge and information that has undoubtedly influenced the talk I'm giving today. At the end of the slides, which you have the link to in the chat, some of the key references that I cited in the talk. Should you be interested in reading more, and you can do some reference snowballing and getting into some pretty deep rabbit holes on proxy system modeling and uncertainties. That's me done.
Thank you very much, Quinn. That was really interesting. So if anyone also has questions, then you can also write them in the chat box. Ramesh said that thanks, Quinn. Great presentation of very interesting aspects to think about. And thanks to the organizers. So I don't know if anyone has any questions for Quinn. And Nikita also says, thanks for the data scientist's take on the paleoecological data. I'm working with from a plant ecology angle.
Great, yeah. And of course, feel free to email me as well. I should have put my email address on the slides as well. I can put it in the chat, but it'll disappear.
Hi, Quinn.
Hello.
I have certain questions regarding your presentation. By the way, thank you. You have a nice presentation. Actually, I'm Sangram Sahu, basically from India. I'm focusing on mangrove ecosystem in coastal area, Sundarban. My question is how you define in coastal ecosystem for your paleoecological context in uncertainty, or what's your point of view in specifically delta region uh, based on the mangrove ecosystem in context of paleoecology and uncertainty.
Right. So if I understand correctly, in this modeling framework, we're actually not trying to recreate a specific environment. We're trying to recreate statistical patterns. Did I say statistical environment? A given ecological environment. We're just trying to recreate statistical patterns. And so if you want to recreate a specific environment, that's where we start getting into trying to develop process-based models and develop actual real mechanistic or represent mechanistic processes in a modeling environment. And that, of course, comes along with its own whole set of problems, because now you end up with these models like actually the one I'm working with now, which is ILAND, where you have hundreds and hundreds of equations going into the model, and many of them, some of them are well parameterized, many of them are poorly parameterized, so you never really know how well you're recreating or how realistically you're recreating a given environment. So in this framework, we've sort of circumvented that, and we're saying, well, it doesn't actually matter how those patterns came about. And this is very much going back to the Blau paper that I cited. He demonstrates that you can recreate all the same statistical properties that we see in an empirical paleo data set with completely random processes. We can have abrupt changes, we can have that sort of sawtooth pattern, we can have species come and go out of the system. So if we can recreate the statistical properties of the data, we can experiment on it and still make a reasonable inference for the effect of those uncertainties on our analyses. So I don't know if that answers your question, but we're not really trying to recreate, say, a mangrove system in this framework.
Thank you. We can connect in, I think, mail through mail, so that we can.
By all means, yeah.
Yeah. Thank you.
Thank you very much. So just before people start leaving, we have another workshop at Quinn who's gonna be held on December 5th. He's gonna show us how to use Bcron. So Bcron is an R package used to HDEP modeling. So I'm just gonna also put like the registration link, so in case you are interested also to attend. So thank you very much for being here, and thank you, Quinn, for this amazing presentation.
Thank you for organizing and for everyone who attended. Yeah.
So thank you very much. I just have a question about, like you said that you want to send some preliminary, like, additional information for like the next workshop so people can, install the package and the stuff.
Yeah.
So I think like we already have some people have a sign up. But maybe I can also share with you like the email list. I usually send another reminder early, like I'm going to send another one next, probably on December 1st, because like some people I was needing, like it's easier
Right.
for them to
Yeah.
remember, oh, okay, it's coming. So and they will sign up. So that's actually when we see an increase in the number of people signing up for the event. But like just let us know, Nick, also how to want to share that information. So we can
Yeah.
also include link to your GitHub account and
Well,
stuff like that.
I'll make a repository with like a README with full instructions to try and install some packages, and hopefully it'll be fairly self-contained. And if anyone has any issues, to like drop me an email so that we can hopefully get all the troubleshooting out the way before doing the workshop. There's always there's always a few issues with whatever R versions or Bcron versions.